Hi, my name is Patrice Witschen, and I am a third-year PhD candidate within the CMB program, working in Dr. Kaylee Schwartfeger's lab. And today I'm going to share with you an abstract from a paper that I recently published titled Tumor Cell-Associated Hyaluronan CD44 Signaling Promotes Pro-Tumor Inflammation in Breast Cancer. As many of you know, breast cancer is the second leading cause of cancer-related death among women in the United States, and therefore this is a relevant disease to study. And interestingly, cancer has been compared to a chronic wound that's unable to heal, as the balance is tipped in favor of pro-tumor inflammation. Therefore, it's important to understand how cancer cells interact with and alter their environment to ultimately support disease progression within this chronic inflammatory state. Now, the tumor microenvironment pictured here consists of both tumor cells and stromal cells living amongst this dense extracellular matrix, and is highlighted in this insert, HA, pictured in these blue squiggly lines, ultimately can surround the bulk of the tumor as well as individual tumor cells themselves. Hyaluronan is of interest because it's this large glycosaminoglycan of the extracellular matrix that is tumor suppressive under normal physiologic conditions. However, if it gets exposed to some sort of cellular or organismal stress and comes into contact with hyaluronidases or reactive oxygen and nitrogen species, it can get cleaved into this low molecular weight fragments, which have actually been shown to be tumor promoting. And before jumping into HA within the tumor, I think it's really important to understand how normal cells make, fragment, and respond to hyaluronan within the context of a wound, for example. So HA is synthesized by hyaluronan synthases 1 through 3, and this high molecular weight molecule is extruded into the environment. It can get cleaved again by these hyaluronidases, for example, and then the accumulation of this high and low molecular weight molecule can then bind to its cell surface receptor CD44, and these HA-CD44 interactions have been shown to promote cell adhesion, produce or lead to the production of inflammatory factors, and then recruit innate immune cells to promote wound healing. So the purpose of the study was to investigate these HA-CD44 interactions within breast cancer to determine whether this axis directly impacts the formation of this chronic inflammatory environment. One of the first things that we showed was that these breast cancer cells are able to synthesize HA. They can also fragment HA into these low molecular weight fragments, and they also express CD44 on their cell surface as illustrated by this flow plot. Additionally, upon knockout of that HA receptor CD44, we showed that in these breast cancer cells, there was a decrease in a variety of inflammatory factors, again, upon loss of CD44 in blue. And so these changes overall are one example of how these HA-CD44 interactions might lead to a change in the inflammatory cytokine production within their environment. Importantly, our lab results were also supported by human breast cancer patient data where increased hyaluronan synthase 2 expression here, illustrated here, is HAS2 high as compared to HAS2 low breast cancer patients. So this increased HAS2 gene expression is associated with an inflammatory gene signature, again, in these human cases of breast cancer. Overall, this work is very significant because a high level of HA deposition within many different tumor types yields or has been shown to yield a poor prognosis. And therefore, our results emphasize that these HA-CD44 interactions potentially have broad implications across multiple cancers. And with that, I'd like to encourage you to go check out the paper in Cancers. And I'd like to thank all of our collaborators and my funding, and I want to thank you for listening.